Hi, everyone, and thank you, Projecta, for the kind introduction. So let me share the screen. So here we have it. Share. Great. So it is a pleasure for me actually to be able to introduce to you the protein proximity network assay. And I will start with a brief introduction regarding actually Pixelgen technologies. I think you can see also my cursor now. Perfect. So if you're not familiar with Pixelgen technologies, at Pixelgen, we study protein interactions, and we do this on the cell surface of single cells. And this is done now thanks to this technology that we have developed, so the proximity network assay, in a manner that has not been previously possible. And this is leading researchers to learn novel insights into disease mechanisms, drug mode of action, response, and also, for instance, biomarker discovery. So during this webinar, I will first give you an introduction to the topic of protein interactomics and looking at the current technologies for proteomic analysis. We will then look at the chemistry of the new assay that we developed, so the proximity network assay, and looking at the key steps of the chemistry. We will then focus on the data output, first with an overview, and then looking at the kind of metrics that you will be able to generate using our assay. So abundance, but also spatial metrics like clustering and colocalization of proteins. And we will also look at some examples of data. A big part of the presentation will revolve around the protocol, looking at the general steps, so an overview in general, and also suggested workflow, looking then at the cell input and sequencing requirements and the kit components, finishing with some application and some examples of application of the Pixelgen Proxiome kit. But let's start with the topic of protein interactomics. So I'm sure you're all familiar with the central dogma of molecular biology, but it is very important to understand the type of components that can be studied in cells. So of course we start with DNA, so the blueprints containing the genetic information from the different components of the cells. Then the RNA, seen as the manufacturing message, and then proteins, important for the cellular functions. And normally when we look at proteins, we look at if a protein is present or not. We look at the abundance level. But the key question here is that, is it really enough just to look at the protein abundance to understand how a cell is functioning? And we know that immune cells require efficient, precise, and direct responses, and this is achieved through spatial reorganization of proteins. So this is why we wanted to introduce a new layers of omics, so the interactomics, which studies the organization of proteins, which is important for regulating cell function. But why Pixelgen? Well, we know that when we look at diseases, these are caused by aberrant cell function, which we have seen is caused by different spatial organization of proteins. And this interactome is very dynamic. It represents a vast and a studied mechanistic dimension in translational research. So for instance, with clinical samples, which has a massive potential as novel types of biomarkers, drug discoveries, and also new therapeutic avenues. And this can be studied now, this protein interactomics, with our assay, so the proximity network assay, and with this kit that we are now able to provide you. So if we look at the different technologies for proteomic analysis of cell function, traditionally microscopy has been the method of choice. And the big advantage of microscopy is, of course, resolution. In case of super resolution microscopy down to 20 nanometer resolution. Of course, the drawback of microscopy technologies is the limited number of markers that can be studied simultaneously. So on the other hand, if you're more interested in multiplexing, technologies like flow cytometry, mass cytometry, CytSeq can help you with that. But in that case, you're going to lose the spatial resolution. So there is this gap here that we wanted to fill in when we developed the proximity network assay, which has very high multiplexing, 155 targets, and very high resolution, 50 nanometer resolution. You will be able to get information from 50,000 spatially localized protein molecules per cell, and very high throughput. So all the markers in 1,000 cells per sample simultaneously. We will also offer free software, which is called Pixelator. I will go back to this one later, but this free software allows you to perform straightforward analysis, so looking at abundance, but also spatial metrics like clustering and colocalization for each protein in each individual cell. Now, how are we able to have actually this very high resolution with this very high multiplexing without having the drawback of microscopy, for example? And the answer is that we do not use light actually to read and see where proteins are localized on the cell surface, but our method is a sequencing-based method. And what we offer is a sample prep kit. So you will be able to work with single cells that are in suspensions in PCR tubes. And you will also be able to identify cell-to-cell complexes. These cells are first fixed, and then you will be applying the reagents that are included in the sample prep kit. During the process, you will generate a PCR product that will be fed into the sequencer, and after into the Pixelator software for data processing and analysis. And finally, you will be able to reconstruct in 3D single cells, like this example here, where you have this cell spinning, and in red, you can visualize the location of a protein of interest, or you will also be able to identify proteins that are cells that are interacting, like in this case, a B and a T cell. So I'm going to show you now a schematic video with the main principles of the technology. So here we see a cell illustrated with the proteins on the cell surface of it. So we first add the antibody panel, so they bind to the respective targets. And then on each antibody, there is a unique DNA oligo sequence. We bind padlock probes, we copy the information from this oligo, and we use rolling circle amplification to amplify this oligo, as you can see here now in the video. We then add linkers. The linkers connect and link rolling circle products which are in close proximity. And we do that for all the 155 proteins that we have in the plex on all surface of the cell, creating this complete network of information. So here you're able to see the different steps of the technology more as a workflow. So as I said, cells are in suspension in PCR tube. There is no compartmentalization of the cells, so everything is in bulk. So cells are floating in suspension in the tube. The first step is a PFA fixation step, done in order to immobilize the surface proteome. We then go through a second step that you have seen in the video, where we bind the barcoded antibodies. We then add the padlock probes. They bind to the oligo attached to the antibody, and we copy the information. And we perform rolling circle amplification to create repetitive copies of the oligo. Basically, we elongate the oligo. We then add linkers. So the linker connects the rolling circle products which are in close proximity. Then off the bench, we perform NGS sequencing, and our technology is compatible on all Illumina sequencers. And after sequencing, you can see here some reads that contain the information from proteins that are in close proximity with each other. We then input this information on our software, Pixelator, to perform data analysis. But how are we able to go from the first basic step, so after fixation, down to be able to create actually in 3D the cell and the spatial organization of the proteins? So actually you can see here, now you can see a bit more the details of actually the oligo. So each oligo, after the barcoding antibody binding, so each oligo contains a protein ID, which is of course unique for each protein species. But then we have a unique molecular identifier. This is what makes unique each oligo. Here you can see the padlock probabilization step where we copy the information from this oligo, so from the protein. And then we perform rolling circle amplification. So we elongate the oligo. And then as you can see here, this is a zoom in where we link rolling circle products that are in close proximity. So you can imagine that since the rolling circle product is creating by repetitive copies of the oligo, one protein can be connected to multiple proteins at the same time. And this is what happens in details in the last step. So what you have here to the left is the rolling circle product from one protein, here the orange one. To the right, another protein, so the green one. As you can see here, we have a first sequencing handle. We then copy the information from the rolling circle product, one. We have the linker that links the two rolling circle products in close proximity. and then we copy the information from the other protein, and we finish then with another sequencing handle. So this is how the final amplicon looks like. We then perform sample indexing PCR to bind sequencing adapters, and then the sample is ready for Illumina sequencing. So this is what happens in a real case, more or less, where you see here 3 different proteins that are in close proximity, CD3, CD45, CD8. After NGS sequencing, these are the different reads that we will be able to observe. They contain information from the 2 proteins that are in close proximity, as well as the unique molecular identifier. So our software is able to take these informations and reconstruct and understand that these proteins were in close proximity, and then place them on the surface of the cell. But what would happen if actually, instead of having 3 different proteins, we have 2 proteins that are from the same species? For instance, we have 2 CD3s. And we have this example here now with CD3, and CD45. These are the reads that we will get after sequencing. And as you can see, there is a read that contains the information from these connections between the 2 different CD3s. Now, in this case, the protein ID will be the same, since this is CD3 protein, but the unique molecular identifier will be different, since this is unique on each oligo. And this is what allows our software to be able to reconstruct this triangle here and place it on the cell surface. So now you are more familiar with how the chemistry works. So let's have a look at the kind of output, the different outputs that you will be able to generate using our technology. So after sequencing, the output of the sequencing is, of course, a FASTQ file. And these FASTQ files are fed into the data processing software, which is called Pixelator. And it's free, and it's available on our website. We have a lot of tutorials you can check, and it can be run with R and Python. And there are also example datasets that can be downloaded, and you will be able to play with the data to have an idea of the kind of rich dataset that you will be able to generate. So after having fed the Pixelator with our NGS and FASTQ file output, you will be getting 2 different outputs from the software. First is a WebQC report, which contains the basic information regarding the statistics of the run, for example, and then the pixel file. So the pixel file is what you will be needing for performing the analysis. As you can see here, some plots that we will see now in the next part of the presentation. And what are the different data outputs that you will be getting? First, as other technologies, you will be able to look at the abundance. But then you will also be able to look at spatial metrics like clustering of one protein, also colocalization of pairs of proteins. So regarding abundance, what we have here, it's a PBM sample. We have performed high dimensional clustering, so we can see here all the different subpopulations. And in the plex, we have also included some contaminants for like platelets or granulocytes, for instance. We have also marker for stem cells and cancers like myeloma. But the beauty of the technology is that we can go beyond this. So we can go into each of these subpopulations here. We can extrapolate single cells, and we can look at the protein abundance and distribution of all the 155 targets that we have in the plex. In this example is a naive CD4 T cell where we look at abundance and distribution of CD45. And here is an effector memory CD4 T cell where we look at abundance and distribution of CD2. But we can do this for all the 155 targets that we have in the plex and for all cells that we have input. So here you will be getting then, you will be able to look at the abundance. So here you have all the different proteins, and then here you have the different cell types, and these data are normalized. This is what you will be able to get. You look at things like exhaustion, for example. But let's move to the spatial metrics. This is what is very interesting, and this is the real power of our technology. So to measure the clustering of a protein, we have introduced what we call a clustering score. So clustering score would be positive if the protein is polarized on the cell surface, as you can see here in the drawing, and will be close to 0 if the distribution of this, if this basically protein is dispersed on the cell surface. This is based on spatial statistics. So basically what we do, we look at the graph network for each single cell, and we look if a protein is close to itself more often than predicted by random chance. And to show you how this works in a real case scenario, we look at the antibody drug mode of action of rituximab, which induces the clustering of CD20 on B cell cancers and enabling the binding of NK cells to initiate cell killing processes. So when we look at the protein clustering for CD20 on the stimulated cells, or cell treated with the antibody, we can detect higher clustering score compared to the control, as well as we have here a negative example where there is no difference between the control and the stimulated sample. And then this was validated with fluorescence microscopy. Here we have the control, so CD20, which doesn't show any kind of clustering. And then we can see on the CD20 rituximab treated cells where we can see actually the clustering of the protein. The other special metrics is the protein colocalization. So also here we have introduced a score, which is called colocalization score. And in this case, we are looking at pairs of protein. So if we get a score close to 0, that means that they are randomly distributed on the cell surface. If we get a positive score, that means that they are actually colocalizing with each other. Also very interesting is a negative colocalization score. That means that the two proteins are segregating from each other. And this actually happens more often than we might think. One example is cells during migration, immune cells during migration, which form a front and a back end, and then we can see segregation between proteins. So this is a pairwise measurement. So we will be able to start looking at protein complexes. But if we think that we do this on 155 proteins that are included in the plex, we will be able to identify actually multi-protein domains. And there are multiple ways of looking at this and visualizing it. One way is creating colocalization heat maps, for example. So here we have a subset of these targets. And what you see here, the circles, these are the colocalization scores. So for example, if we focus here on HLA-BC and B2M, we see colocalization score, positive. Of course, this is a well-known protein complex. What you see here on the diagonal, this is the colocalization of a protein with itself. And then when you start visualizing here, all these squares, these are the multi-protein domain. So this is a B sample, for instance, that's why we have the B cell receptor. We have, for instance, here that there's a Spanish domain. And if you're interested in particular in a certain protein, for instance, CD19, you will be able to identify all the different interaction partners with that protein of interest. So I hope that this part was clear. So now we know a bit more regarding the chemistry of the technology and also the kind of data output and the rich data sets that you will be able to generate. But let's have a look at the protocol. So the beauty of the protocol, and these are the general steps, is that there are no specialized equipments which are required. So the protocol has a majority of hands-on time. Most of the time, the cells will be pelleted at the moment of your PCR tube. You will be adding reagent mix, resuspend the cells, and incubate in a standard thermocycler. You will be then adding washing buffer, perform centrifugation, discard the supernatant, and start again, either with more washes or by performing other incubations. So there are many incubation, many washing steps, but the protocol is quite quick, as we will see in the next slide, and there is no specialized equipment which is required, so you will be able to do it in your own lab easily. So here is to have an idea of the different steps. I mean, I already described them during the workflow. Here it's a bit more in detail. You can also appreciate the duration of each step. For example, the cell fixation, which is the first step in order to immobilize, again, the surface proteome, is divided in 3 steps and takes about 2 hours. And you can also see that there are multiple stopping points in between each step to add flexibility to the protocol. The barcoding antibody binding step goes overnight, the incubation. The second day, you will start by adding the secondary antibody binding, which stabilizes the primary. And then there is the protein proximity network generation with all the steps that we have seen earlier with multiple stopping points. This takes about 5 hours. And the last day will basically be the PCR amplification part, the sample indexing, and the NGS preparation. So quality control, quantification, as well as the pooling of the libraries for sequencing. So let's say a suggested workflow for 3 consecutive days will require approximately 2.5 days. So the first day with the cell fixation and the barcoding antibody binding that goes overnight. The second day starting with the continuation of that step, continuing with the protein proximity network generation. And the last day with the PCR amplification, the sample indexing, as well as the NGS preparation. But as we have seen before, each step has stopping points for flexibility. Cell input. So here you can have an idea of the cell inputs and the counting requirements for the different steps of the protocol. So we require at least, this is a supported range, at least 500,000 to 1 million cells for the fixation step. And so here you will require cells in suspension. For instance, if you work with French frozen tumor biopsies, you will need to perform dissociation. But this is what we require for this step. It's important to avoid losing too many cells for the next then steps. For the barcode antibody binding, the supported range goes from 50,000 to 250,000. Here you can see the recommended cell input. Protein network generation from 20,000 to 100,000 cells. And then to PCR, the input that we recommend having to have enough statistical power is 1,000 cells. Regarding the sequencing recommendations, we have two different sequencing depth. For PBMCs, 250,000 reads per cell, and for cell lines, 500,000 reads per cell. And you can also see the read configuration here. All these informations regarding the cell input, the sequencing requirements, and also the workflow of the protocol, you can find them in the user manual. You can download it from our website, and there is also a QR code at the end of the presentation that you can scan that and you can immediately check then the protocol there. And we also require 15% of 5x, for instance, and the low concentration. We recommend to follow Illumina recommendation for 100% 5x library. So this is how the kit looks like. So there are three different boxes. The first one, which is sort of +4, which contains wash buffer, antibody panel, secondary antibody, and the quenching solution. And then two boxes as -20. One which contains all the reagents needed for the protein proximity network generation, and the other one that's -20, which contains the primers. So as you can see in this example here, you will be able to process 8 samples at the same time. We do offer, though, 3 different unique sets of primer, enabling the sequencing of 24 proximity network samples in the same flow cell. What are the applications of the technology? So I'm not going to go into each of the applications. This is going to be more an overview of the different applications where the Proxium kit has been applied. So we applied it in drug development and pharma to discover biomarkers of drug response based on protein clustering. Also, we have seen an example with the rituximab to differentiate the mode of action of antibody drugs, for example, in therapies, by their effect on the cell surface protein dynamics. In hematology, ALL, AML, where we look at stratification biomarkers based on protein clustering, molecular mechanism of drug action and resistance by protein interactomics, and also looking at novel therapeutic targets. Big field of research is immunotherapy and cell engineering. This was used to differentiate the molecular mechanism of action of CAR-Ts, nanoscale mapping of CAR protein neighborhoods, CAR-T trogocytosis, and also quantification of cell-to-cell complexes. It's been used in basic immunology for T-cell migration, differentiation, activation, organoids models, and also for discoveries based on deep phenotyping, and also trauma response, brain injuries, vascular trauma, and recovery studies. So as you can see, very broad applications. And what we always say that this is a tool for discovery. So we are very interested in knowing more applications where this kit can be applied. And this is what you will be able to generate in the end. So this is a real T-cell data. And what you see here, of course, this is a projection of the cell. You can see highlighting all the different markers that we have in the plex. And again, up to 155 targets can be visualized simultaneously on the cell surface. So to recap, regarding the Pixagen Proxium kit, so what we offer is 155 protein markers for sequencing-based protein interactomics. There are 8 reactions per kit, up to 1,000 cells per reaction. And what we offer is a sample prep kit and a free software. And again, I already said during the presentation, it's very important to stress this concept that there is no specialized instrumentation that is needed. So if you want to know more about the chemistry, you can check our website. We have also different videos there. You can also always email us at hello@pixagen.com to have more information. The user manual is available online. You can also scan the QR code here. We already started shipping the Pixagen Proxium kits. So if you are interested, you can contact our business development managers. You can see here the names here and also the region where they operate. And if you are curious to know also the different targets that we have, so the 155 proteins, you can download the list from our website. And today's webinar was more focused on the chemistry of the technology, the kind of data output, and also looking at the protocol. Next week, we will have another short webinar, which will be held by Hanna von Hoyen, and she will be mostly talking about applications of the chemistry. So the title of the webinar is Dissecting the CAR T-cell membrane using the proximity network assay. So feel free to register. And then we have also recently published an app note on our website, and you can scan this QR code to know more about the CAR T applications of our technology. And you can always contact us to organize seminars at your institution, or you can contact also the BDMs that operate in your region for that. And with this, thank you very much for your attention, and I'm happy to take your questions.
Thank you so much, Michele. Let's give them some time to write their questions. Maybe I can ask you a question in the meanwhile. So what are the different options for sequencing?
So as we saw during the presentation, as of today, the technology is available on all Illumina sequencers. So it's compatible with Illumina sequencing. It might be that in the future we can open for other technologies, like for instance, the ABT system of Element Bioscience with minor modifications. But for now, the technology is available on all Illumina, it's compatible with all Illumina sequencers.
All right.
And I mean, just to also, and if you want to know, I mean, we have seen a bit of the reconfiguration, all these kind of informations. Everything is available, as I said before, on the user manual. So feel free to of course check it, and you can also email us if you have specific questions.
Yes, the user manual, application note, they are all available on our website in the resources page. I also wondered what kind of cells can be profiled with our technology.
Yeah, so for now, and probably for some time, the main focus will be on immune cells. And I would say because probably like this, we are, as you have seen also in the application slides, we are able to cover multiple fields of research. And the immune cells are involved in most of the, if not all the disease. So what we have now tested is, of course, PBMCs, as you have seen in the data. We've also worked with bone marrow samples, freshly dissociated solid tumor, and also where we, for instance, in that case, we look at immune infiltrates. And we have also applied it on cell lines, for instance, when we look at different co-culture experiments. And for instance, you can find this more on the app notes.
All right. We have a question here. How challenging is it to analyze the data computationally? Is it straightforward? How does it work?
Yeah, so this is a very nice question. So I think it depends a bit. If you are used to work with single cell data, probably I don't see that many differences. And it is not that much more challenging compared to that. It might require a bit of experience, I would say, for instance, of running Python or R. But we have a lot of documentations on our website regarding the Pixelator software. And you can also download, actually, data sets if you want to look at the data and have an idea how challenging it is to work with the data. We have also bioinformatics scientists that can help you with this, and can help you in, for instance, performing analysis, parallel analysis, and giving you the support that you need to work with your data.
Excellent. So if you don't have any more questions, then I'd like to thank you, Michele. Thanks for today's webinar, and I hope to see some of you in our next webinar. That's about it. Thank you everyone for joining.
Thank you. Bye-bye.
Bye.